Swayam Prabha. Digital India. Educated India.
Welcome you to the next class on the Inorganic Chemistry of Life, Principles and Perspectives. In the last three classes we have looked at the enzymes based on the nickel, which really contributed to the life genesis itself, going from a carbon monoxide atmosphere which is reducing atmosphere to that of the one what we enjoy today. Now we move on to next set of enzymes. So enzymes based on the copper. So let us look at the biological and organic chemistry, the copper enzymes. Before we go more into the details of the copper enzymes, their functions, let us look at a bit information on the classes, various classes of these copper proteins or copper enzymes. One of the major class of these particular copper proteins is called blue copper proteins, and these are blue copper proteins because they are blue in color. I will explain in a while why they are in blue color. And the main function of these proteins is the electron transfer. The variety of such things are there like azurin, plastocyanin, etc. I will come back to that in a while. So they are found in algae, green leaves, plants, many kinds of systems that we have. And then we also have another set of class of enzymes called blue oxidases. These blue oxidases contain one of these blue electron transfer protein plus some oxidizing center as well. So these are there, okay. So both of these. So these are like lacase, like ceruloplasmin, ascorbate oxidase, etc. So these are found in fungal, animal, human, plants, all kinds of things are there. So these have got, one of them is the electron transfer blue protein where the blue color is coming from, and then the redox center for the other part of it. So this can be involved in a variety of reactions, oxidations of phenol, okay, and ascorbic acid, many of these things, okay. So and some of them are also involved in the transport phenomena too. We will come to that little later. Let us see another class of enzymes of copper which are referred as copper based superoxide dismutase, and we have studied several times the superoxide dismutase enzymes. Their main function is to remove the O2 minus dot, which is superoxide. So detoxifying the superoxide, these are present in the red blood cells, and you would see that this is a bimetallic enzyme with one copper and one zinc, where the copper is a functional, zinc is non-functional, rather structural. We will get into all of those. Then we also have another class of proteins, copper proteins, where cytochrome C oxidase, which is obviously mitochondrial one, and this is nothing but a terminal oxidase of this. Besides this, we also have non-blue oxidases. That means there is no blue copper protein present in this, okay, and these are called non-blue copper oxidases. This reduces O2 to H2O2, and this amine oxidase, galactose oxidase, these are some of the examples of the non-blue oxidases. So this means that they do not have the blue color centers that were there for the earlier case. And these are found in animals, moles, etc. So, and they are involved in the collagen formation, elastin formation, oxidizing the galactose. So many of these kind of reactions. Come to another class of proteins, which is the oxygen carrier. We have already seen the oxygen carrier proteins earlier, and we have also combined and compared between the hemoglobin and hemocyanin. So there we have seen a di-copper center, and these are involved in oxygen transport, and these are present in the molluscus and arthropods, and we have said at that time, because of the presence of these ones, their blood is blue, and we looked at all these when we were talking about the oxygen transport by the protein of hemoglobin systems, and we compared as well. So, and you also have, like in the iron, monooxygenase and the dioxygenase. Monooxygenase, so that will add only one oxygen, and the dioxygen will add both the oxygens of the O2, and some of the corresponding things are shown here. Tyrosine oxidation, and it will give some form of the oxidized, which will further convert into melanin. Okay. So, dopamine to norepinephrine. So, this happens in the brain of the human. Okay. And for dioxygenase, quercetin oxidative cleavage, these are present in the fungal. So, now we have seen a large number of different kind of enzymes having blue copper protein, simple electron transfer with oxidase properties. Then we have seen non-blue copper and with the oxidase properties, and we also have seen some where all these together are present. So many kind of different kinds of the things. So, here we see that a few additional other kinds of things. So, amino acid oxidase, catechol oxidase, dopamine monooxygenase, lysine oxidase, peptidyl glycine monooxygenase, lysyl oxidase, metallothionein. So, variety of these things are there in the copper enzymes. So, it is going to be a very huge story, then we need to go try to understand one by one its properties, etc. Now, what will happen when you have, in the beginning stages I have explained metal ion need and deficiency and excess kind of a that is dosage deficiency excess kind of syndromes. Now, if you look at the deficiency of copper can lead to the decreased circulating copper levels, then will make the iron deficiency also, and that will, if it leads to the ceruloplasmin, this is the kind of a function. It can lead to the deficiency of building cytochrome C oxidase, then that will lead to hypothermia and muscle weakness. If the copper deficiency leads to dopamine beta hydroxylase deficiency, then hypothermia, neurological defects like that. So, that means a number of copper based enzymes will be affected as a result of the deficiency of the copper ions. So, this enzyme can get affected, this. So, all of these will have corresponding. For example, if the copper based superoxide dismutase enzyme is affected because of the low concentrations of the copper in the cellular concentrations, then what will happen? This enzyme will be low in concentration. If this enzyme is low in concentration, it will not be able to protect the cells from oxidative stress. So many things are possible. So, all this is because the ion is deficient, therefore, the corresponding enzymes are not being developed. As the corresponding enzymes are not developed, the corresponding functions are not shown. So, this is how one needs to attach one to the other. Okay, so let us look at these the blue copper proteins, and these are also referred as type 1 copper proteins. Type 1, this one has nothing to do with the copper oxidation state 1. It has nothing to do with the copper 1 oxidation state. So, type 1 copper proteins, which are nothing but the blue copper proteins, I said they give intense blue color, and simply the blue color is coming because of a charge transfer. The charge transfer from cysteine sulfur to Cu2 plus. So, this is the kind of thing where you find, and the in this kind of a proteins, the copper site is distorted, either distorted tetrahedral, distorted 4-coordinate or distorted 5-coordinate trigonal bipyramidal. So, and they show, depending upon these, unusual EPR properties. So, you have different kinds of subunits into these plastocyanin, pseudoazurin, amicyanin. All of these are called the blue copper proteins. And the azurins, amicyanins, pseudoazurins, plastocyanins, they have a similar kind of a copper coordination. So, their coordination is 2 histidine, 1 cysteine, 1 methionine. I will come into more details of the coordination sphere bit later, after I cover type 1, type 2, type 3 and other types of protein. Then I will come explain you the actual coordination transfer. So, what is the function of these kind of proteins? The main function of these proteins, type 1 copper proteins, is electron transfer. So, we will see how they do the electron transfer, etc., etc. Now, having looked at some general features of type 1, let us look at the general features of type 2, then we will choose type 3, we will see combined types, we will see all new kinds of types, then we will go into details of each one of these. In the type 2, you have a kind of a close to a square planar or a square pyramidal kind of a coordination containing oxygen and nitrogen, no thio group. There is no thio is involved, therefore, there is no charge transfer, therefore, there is no blue color. So, these are non-blue copper oxidases. So, the copper 2 is EPR active with a normal signal and there is no intense blue color at all. So, copper zinc superoxide dismutase, dopamine monooxygenase, galactose. I have shown you some of these examples in the table that I have given you couple of slides before. So, galactose oxidase, copper containing amine oxidase, all of these will contain type 2 copper, type 2 copper, type 2 copper proteins. These are all referred as. In some of the cases, it is not only the side chain of the protein is binding, there is a modified version of the side chains of the protein, they act as a cofactors. So, I will again explain this when I come to the story of the type 2 copper proteins. Okay. Then type 3 copper proteins, these are not from one single copper ion, these are from 2 copper ions, it is called dinuclear copper center, and these are, there is no EPR activity. This is referred as type 3 and this is found in a number of enzymes like tyrosinase, catechol oxidase, etc. alone in tyrosinase along with other centers in the catechol oxidase and the other kinds of enzymes too. So, this protein contains a metal center which is a structural analog of the hemocyanin. In a hemocyanin you found di-copper center, here also you will find di-copper center. Okay, multi-copper oxidases where it will have type 1, we have type 2, it will also have type 3 together. So, that is what is called multi-copper oxidase. Their function is oxidation, redox properties of this, and there are several kind of coppers, of which one is type 1 which is being blue copper protein, type 2 non-blue center but oxidase property, type 3 is a di-copper center, and all these three can also function. So, these are all present in ceruloplasmin, lacase, ascorbate oxidase, all of these have got type 1, type 2, type 3 centers, they function in a coordinated manner. Okay. So, for example, here ascorbate oxidase, a trinuclear copper site is present. So, consisting of a type 3 copper site which is very close to the type 2 copper site, therefore, it became 2 plus 1, 3 coppers. Then far away from this there is a type 1 also, far away from this, which is about 15 to 20 angstroms away from this you have a type 1 also. So, the ceruloplasmin functions as ferrooxidase and the iron 3 produced in this reaction can then oxidize the same substrate as lacase. So, you do not need to so much worry about this point of it. So, all that you need to understand from this particular slide is that you have a type 1, you have a type 2, you have a type 3, you have enzymes where all of these are also present together. Whenever type 1 is present, it will have the blue color, okay, even if other things do not give the blue color. Okay, there are other special type of copper centers are there which are present in cytochrome C oxidase. Okay, and these are referred as copper A, where one of the copper is isolated metal ion referred to copper A, and some places it will be written as copper small d is a capital A. The second copper center is a part of the dinuclear center with cytochrome A3, and this is referred as copper B, CuB, okay, CuA3 is all. So, CuA, CuB are the most frequently used terminology. So, there is a striking similarity between the two of the copper centers in the N2O reductase, nitrous oxide reductase. We will not look under the copper system, we have already looked at under the iron case, okay. So, we will not look at that, but just say that there is a similarity between the copper centers, the N2O reductase and the copper A. Copper A is present in the cytochrome oxidase. Copper B is also present in the cytochrome oxidase. So, copper A center, copper B center. So, these are special centers besides the type 1, type 2, type 3. So, all these we have, and you can see there just the simple structures of these. The type 2, as you can see there, it has no thio group, histidines and some other groups, okay. They could be 4, they can be 5, in some cases 4 coordinate, some cases 5 coordinate. And type 3 has got this kind of thing, hemocyanin, we have already seen, this is oxidized hemocyanin. Blue copper oxidases, where this is a type 2 type and this is a type 3, and there is one type 1 away from it. So, but this is coupled, so type 2 and type 3. So, this portion is type 2, this portion is type 3, it is together. Okay. And now we are talking about type A, B. Type A, B types are here, type A in cytochrome C oxidase and type B in again in the cytochrome B oxidase, but away from C oxidase. So, these are all the kind of centers that you have. Now, what we have looked at? We have looked at the type 1, type 2, type 3, copper A, copper B, and we have not looked at the copper Z. There is another one, copper Z is also there, which is a tetranuclear. Copper B is from the dinuclear case. So, these are some characteristics are given. They all have absorptions in the more or less in the visible region, and the absorptions go from 450 to 700. So, 450 to 480 to 530 to 640 to 700. So, that means your coordination cores are changing from one to the other, their emission spectra are shifted into the visible region in these ones, you can see. And the type 1, the 4-line EPR, type 2 4-line EPR, type 3 no, you cannot detect non-EPR. Copper A has 7-line EPR, copper Z has 2 into 4 line, because that is the, further coupled into that line spectrum. So, that is referred as a 2 into 4 line spectrum. So, you have initially 4 lines, each of the 4 lines, each of the line is further divided into 2 lines. Okay. So, the common kind of ligands are what? Histidine, cysteine and methionine in this case, and here there is no cysteine in type 2 and there is no cysteine in type 3 too. So, you can see that. Copper A has got and copper Z has got sulfido. So, they have the distorted tetrahedral, in some cases trigonal, bipyramidal, distorted, tetragonal, distorted tetragonal, all these kinds of geometries you can go through, okay, all these examples. So, now we got familiarized with type 1 and with type 2, with type 3. Type 1, type 2 are mono copper center, and type 1 has got not only histidines, it has got one methionine and one cysteine. That is the reason why you get the blue color into these ones, and then you have the type 2, no cysteines, no methionines, no blue color, but still one copper center, and type 3 the 2 copper centers that you have, and no cysteines, no blue color from this, and then the one 4 which is having all 3. Now, we are referring it as a type 4. You see these 2 coppers are referred as type 3, this copper is referred as type 2, very away from it, which is called the type 1. So, it has one type 1, one type 2, one type 3. These 2 are within 3 to 4 angstroms, and this is about 15-20 angstroms away from this. and the role of this is, we know very well, it's electron transfer, and this is redox, oxidative reductive type, and this can even activate the dioxygen too. And here substrate can bind as well. So, electron transfer, O2 activation and substrate binding, all three together in one, so it is called oxidases. Now, the other thing, just lately we have looked at copper A center, copper B center, both of these are present in cytochrome C oxidase, and copper Z center, where 4 copper centers are there, and this is where you have the 4 copper centers, these tetranuclear copper Z center is found in nitrous oxide reductase, okay, and the 4 copper atoms are coordinated by 7 histidines and bridged by a sulfur atom. You can see that the one here, that is the sulfur atom here, and these are the 4 coppers here, and there are histidines are bound to this. So, this we may not be looking into the more details of the thing, because these are not very well understood in terms of the mechanism, but they are there very well. Now, what you have seen, you have sufficiently large things that you could see. The copper is involved in a variety of enzymes, type 1, type 2, type 3. Type 4 is nothing but summation of type 1, type 2, type 3, and copper A, copper B and copper Z. So, these are the things. So, type 1 is blue copper, type 2 oxidase, type 3 it can activate the O2 and it is a dicopper center. Type 4 has got all these type 1, type 2 and type 3, all the 3 together, as you can see here, and all the 3. Type 2 is here, type 3 is here, type 1 is here, and copper A and copper B, which are in the cytochrome C oxidase, and this is the copper Z. Okay, having looked at the general classifications, general type of copper centers, let us go more specific. Let us take to start with type 1 copper centers. These are also referred as blue copper centers. These are also referred as electron transfer copper proteins. Whether you say electron transfer copper protein, whether you say blue copper protein, or whether you say type 1 proteins, all of them are one and the same. So, do not get confused. So, I will be using different terminology at different stages. So, type 1 is same as blue copper protein, is same as the electron transfer copper proteins. Now, you can see one of them here, active site of that, other one active site of that, other active. So, what we need to see, the one in the middle, you have 2 histidines, one SME methionine, and this is the glutamine, and here 2 histidines, one methionine, one cysteine. This will be very strongly blue color, and all of these because of the charge transfer from copper 2 plus thio to the copper 2 plus center. So, this is a 5 coordinated is roughly trigonal bipyramidal, but distorted. This is distorted tetrahedral, this is also distorted tetrahedral. So, you have generally 4 and 5 coordination. So, azurin, and you see that the redox potentials are changing, 285, 185, 255, 375, 260, 680. So, that means the lowest here is 185, the highest here is 685, around 680. So, the difference is about 500 millivolts. So, by changing the coordination, of course, is not sufficient enough, and the protein also probably must be making an absolute contribution to the redox potentials of this. So, what is the advantage of having so much different, so that you can do variety of redox reactions using this one. So, electron transfer can be transferred from one to the other to the other to the other very efficiently when you have different redox potentials that you have in this enzyme. Okay. So, you can see that. Now, the same thing, let us look at the enzymes. Let us look at the enzymes. So, we have looked at the enzyme here. This is an enzyme, and this is the copper center. This is an enzyme, this somewhere here is a copper center. This is an enzyme, somewhere here is a copper center. As you can see that somewhere here is a copper center. So, you can see here somewhere here is a copper center, somewhere here is a copper center, somewhere here is a copper center. So, in all these proteins, protein has a lot of structure, and towards roughly towards terminal, some of them are a bit more exposed than the other. They are all towards the end of the protein. So, this protein roughly can be taken as cylindrical, and towards the end you have this copper center. So, this is a protein with 680 millivolts, 375 millivolts, 285 millivolts, etc., etc. One example you can see here, this is your copper, this is your S-methyl, and this is your cysteine, this is one of the histidine, this is the other histidine. So, 2 histidines and 1 methionine, 1 cysteine, and the remaining protein you can see here. Very nicely you can see the blue protein here. Okay, if you look at the closely these ones, you can see the coordination sphere very nicely, very nice, very good, and very good here. So, you can see all these coordination spheres. So, either the highly distorted, you can see the very highly distorted tetrahedral, also highly distorted trigonal bipyramidal. You see why highly distorted? You can see this SCUS angle is very, very acute kind of an angle. So, all these kinds of things there. So, the same thing is shown here, which of the residue is binding. So, this is not very essential, you can always look at whenever you think like to. This I already explained to you. Explained to you that the redox potential change a lot, though the coordination changes only somewhat. So, that means both the coordination is contributing to this redox potential, as well as the protein conformation is contributing to the redox potential. So, if you see close by this copper 1, one of the copper 1 cent type 1 case, this is azurin, this is the trigonal, this is the bipyramidal kind of thing that you can see highly distorted. And you can look at plastocyanin. You see this copper to sulfur bond, thiolate bond is 2.85, 285 picometer is nothing but 2.85 angstrom, which is very far away, but forms a coordination. So, therefore, such kind of a distortion, why such a kind of distortion is nature has chosen, something that one needs to look at that. Yes, we will look at that now. See the copper, this protein, what is its function? Electron transfer. So, electron transfer means it can go between, copper can go between 2 plus and 1 plus. In the oxidized state it is copper 2 plus, in the reduced state it is copper 1 plus. Again back to oxidized, copper 2 plus, back reduced, copper 1 plus. So, therefore, 1 electron transfer. So, it is going between copper 1 and copper 2. So, when you this is from the oxidized protein. Now, when you go to the reduced protein, crystal structure is known. This distance is brought down from 2.85 to 2.7475, and this is moved a little bit farther away, and this is a little bit dropped, this is a little bit moved towards this. So, there is a lot of change in the geometry of this, and these are connected to the protein. So, when the geometry changes here, this will change the protein conformation. So, therefore, the protein conformation is coupled with these geometry changes, which is coupled with redox. So, redox copper 1 copper 2 is coupled with these changes in these geometry parameters, which in turn is converting, which in turn is changing the protein conformation. So, therefore, protein conformations are very much influential in this. Now, why is this so much distorted? As I said, you need to go through a copper 1 plus and copper 2 plus, and as you know very well, the copper 1 plus prefers more of a tetrahedral, copper 2 plus prefers more of a square planar. So, therefore, you have somewhere in between. This particular geometry is neither a square planar nor a tetrahedral, therefore, in between. So, because of the in between, the amount of reorganization required or reorganizational energy required including the redox is very minimal, and that is the reason why the protein has chosen such a kind of a geometry in this too. You can see that. Now, how will it do the electron transfer? It will join with another protein or the receptor or other counterpart of the biological system, and this is from your plastocyanin, and there is a overlap of these things or some other kinds of antennas where the electron transfer. So, the electron transfer I explained to you very well when I was initially introduced topic of the under the iron, where I talked to you for the iron sulfur proteins as well as I talked to you cytochromes. Prior to that I introduced to you the two proteins come closer. The one is giving away electron, other is taking the electron, vice versa. They come, there is a reorganization of this, and then the electron transfer taking place between them, either from the through a inner sphere electron transfer or outer sphere electron transfer. Many times you will have an outer sphere electron transfer, and that is the kind of thing that you can see here too. And so that is where we have the electron transfer part of this we have. So therefore, I have started explain to you the different classes like type 1, type 2, type 3, type 4, kappa A, kappa B, kappa Z kind of things, and I started with type 1 electron transfer, how the electron transfer is facile because of the distorted geometry during the copper 1, copper 2, and the protein electron transfer is very efficient to the other biological proteins. Thank you very much.